Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Wednesday, December 12th, 2012. We begin with a story from the world of nanotechnology. A few weeks ago, we discussed the use of light-activated nanoparticles for the production of steam. Now, researchers at Rice University are investigating something similar for use in medicine. Specifically, what are known as plasmonic gold nanoparticles that heat up when exposed to a laser pulse. When the nanoparticles heat up in a liquid medium, they create small bubbles of extremely hot vapor for a brief amount of time. On the scale of a living cell, it's like an explosion, but with a limited enough range to be selective, which is why the researchers are investigating these plasmonic bubbles as a form of cancer treatment. And in their most recent experiments, they discovered that altering the nanoparticles' properties changes the kind of bubbles formed and their potential function. Most importantly, the two types of particles could work simultaneously. Hollow particles were placed inside cells, and the resulting bubbles destroyed those cells. Other cells in the same medium had solid particles that created smaller bubbles, forming temporary holes in those cells' membranes. Which is important because those pores could allow large drug molecules or DNA for gene therapies to enter. All of this means that these mixtures of particles could be activated with a single laser pulse, killing some cells and allowing others to be modified. It's unlikely a process like this would be convenient to administer inside a patient, but could be incorporated into the outside processing of cells. For example, bone marrow or other stem cells being prepped for transplantation. The patient's own tissue could have cancerous cells eliminated while modifying the remaining tissue to increase regeneration. As the research continues, those working on it will have that goal in mind. Speaking of stem cells, our next story is news from the world of medicine. Researchers at the Salk Institute have developed a new technique for creating stem cells with potential therapeutic applications. For some review, let's go over the basic types of stem cells. There is embryonic stem cells, which actually aren't usually extracted from an embryo, but instead a blastocyst. That's a bundle of cells that results from a zygote dividing for 5 days that has not yet implanted and is technically not an embryo. However, the ethical issues aside, embryonic stem cells could only really be useful for research. For therapeutic applications, most research is going into induced pluripotent stem cells. This is when an adult cell is converted into a very embryo-like state through a variety of methods. These cells have the potential to become nearly any kind of cell, but have their own issues. For one, induced pluripotent stem cells have a risk of turning into tumors, because really, cancerous stem cells actually have similar properties. And two, production can take a long time, which could be an issue if the goal is producing a large quantity of patient-specific cells for a treatment. So, the Salk Institute researchers are taking a slightly different approach dubbed indirect lineage conversion. It's similar to the pluripotent method, but instead produces multipotent progenitor cells. For example, the researchers turned skin cells into the progenitors of vascular cells. These progenitors could multiply and become a limited variety related to vasculature. This process cuts out the pluripotent stage, reducing cancer risk and processing time. It might be years before this process is perfected, but the researchers believe that indirect lineage conversion is a very promising avenue for creating stem cells for use in treatment. Finally, we end with an update from the world of chemistry. As you probably know, increasing concerns for the environment are changing how humans do things. This not only applies to greenhouse gas emissions, but also harmful chemicals used in industrial processes. So an international research team led by McGill University has been analyzing a more environmentally friendly way of producing chemicals. Conventional chemical processing, like the synthesis of pharmaceuticals, generally involves large amounts of solvents and other compounds that are potentially toxic. An increasingly popular alternative is mechanochemistry, which is done mainly through ball milling. Essentially, reactants and catalysts are placed in vibrating containers while steel balls are rapidly shaking around. You might think this is only useful for the breaking apart of materials, but it can actually serve as an effective synthesis method. How mechanochemistry works relates to the impacts between the balls and the container. These impacts create momentary areas of increased heat and pressure that can facilitate chemical reactions, especially with the assistance of catalysts. Although this is the principle behind it, actually observing the chemistry in detail is another challenge, which is why the team went to ESRF in France for their high-energy X-ray source, allowing them to observe the reaction kinetics in real time through the metal walls of the reaction container. So far, they've only observed one kind of reaction but plan to study more, allowing them to optimize the milling process, hopefully leading to the safer synthesis of chemicals such as pharmaceuticals and other complex compounds useful in industry. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.